Thank you, Abby, for the introduction. And also thank you for the company of Biologists for giving the opportunity to present our work and also recognizing our work for the award by the awards committee. So I'm a postdoc in Shankar Srinivasa's lab in University of Oxford at the Department of Physiology, Anatomy and Genetics. And we are interested in early development using the mouse as a model to study mammalian development. So let's see if this works. Yep. So if you can think of embryogenesis as an exercise in breaking symmetry and deriving complexity, starting from very simple origins. And in order to do this, the embryo needs to very intricately weave together two processes, one being the acquisition of form or morphogenesis, where cells need to go to the places where they're needed and the tissues are sculpted in order to give rise to the anatomy of the organism, if you like. And alongside, it also has to undergo the acquisition of function, whereby cells, tissues, organs, and systems are gradually acquiring and specializing to carry out the mechanisms that are necessary to sustain life. So I'm interested in how these two processes are mediated through coordination between cells and collective cell behavior. So just to give you a few examples of collective cell behavior from early mouse development that we are interested in. Firstly, you can see just after implantation, the migration of the anterior visceral endoderm, a group of cells which are defined at the distal tip of the embryo, but then collectively migrates proximally to the embryonic extraembryonic boundary, from where it patterns the underlying epiblast and sets the first anatomical axis of the body, which is the anterior posterior axis. And secondly, gastrulation, which is quite famous, which gives rise to the three germ layers of the body from which all the organs and systems are derived, and also establishes the basic body plan. And also, as the first organ starts to form, you need collective cell behavior in order for these organs to start to function. So if you take our favorite organ in the lab and also the first organ of the embryo to form, which is the heart, we know all these processes are important. Firstly, in defining exactly where the heart is to form, generating the material or the cells from which the heart forms, and giving heart its function. So there's growing evidence to suggest that collective cell behaviors are important and calcium could act as a universal information currency in coordinating these collective cell behaviors. So in this talk, we'll cover a quite wide range of development. But in the first part of the talk, I'll try and focus on these early stages, trying to ask the question if calcium is involved in coordinating collective cell behaviors, starting from fertilization, ending at gastrulation. And in the second part, I'll turn my focus onto the heart and see how oscillated calcium oscillations emerge in the heart and how as the heart starts to function. So we know that life, or if you like, embryogenesis starts with a calcium spark. In fact, the act of fertilization induces a wave of calcium that traverses across the egg, which converts the egg into the zygote, or otherwise known as egg activation. In the mammal, this wave takes about 2 seconds and is followed by periodic calcium responses. This has been known for a long time, but little is known about calcium dynamics of the embryo soon following this. So what we did in the lab was try to investigate this in the mouse embryo, combined the GCAMP calcium reporter line with fast time-lapse microscopy in order to see the calcium dynamics during development. So what we found was after the initial fertilization wave, calcium activity in the pre-implantation embryo is quite silent. No cellular oscillations are seen, and if any, they are very rare and stochastic. However, the picture completely changes with implantation when you start to see frequent and widespread calcium activity throughout the various tissues of the embryo. This includes calcium oscillations in individual cells of the visceral endoderm, which is the tissue layer surrounding the embryo, and also in the extraembryonic ectoderm. And you also see rare events such as calcium waves that propagate through multiple cells of the epiblast. So then this is just a mid-central section through the embryo, but to investigate this in more detail in 3-dimension and at faster time resolution, when a very talented master student, Konstantinos, joined the lab, we employed lattice light sheet microscopy to image it in 3D and at high temporal resolution. And these are just highlights of few findings that we got. For example, we found out that the epiblast waves, in fact, are very widespread and it involves a large volume of the embryo and sometimes even occupying as much as 2/3 of the whole epiblast. And these epiblast waves were usually triggered by a VE cell. So a VE cell that lights on before the wave, and actually it outlasts the epiblast wave. And so this was interesting. So these are both single epithelia, and there's a basement membrane between the VE and the epiblast. And both of them abut each other using their basal surfaces. However, we also found that the epiblast wave propagates from the apical to the basal. So these are interesting revelations from fast imaging that we're trying to investigate the mechanisms for. We also found out that calcium oscillations are not reliant on extracellular sources of calcium or calcium release from lysosomes. And although I'm not showing the data here, we found that it is dependent on calcium stores within the endoplasmic reticulum. So we were now able to target these calcium oscillations and ask if these calcium oscillations have any biological significance. So we can pharmacologically stop the calcium oscillations by targeting the calcium reuptake channel of the ER. And what we see is when we culture embryos in control conditions, for example, during AV migration, the cells that start at the distal tip over a 6-hour period can migrate towards the embryonic, extraembryonic boundary, from where it patterns the epiblast and sets the AP axis. However, in the absence of calcium oscillations, the AV cells are stuck at the distal tip, so correct axial patterning doesn't happen. So a lot of this data has been now deposited as a preprint on bioRxiv, and you can all go and have a look at it. And just to summarize that initial part, what we saw is that from implantation onwards, calcium oscillations can be a means by which cells communicate in order to drive this coordinated behavior. And one of these behaviors that we found that it's important for is AV migration and setting above the axial AP axis. So once AV migration is complete and the AP axis is specified, the next step is gastrulation. So on the opposite side, at the proximal side, the epiblast cells undergo pluripotency exit, delaminate, and ingress through the primitive streak to give rise to the mesoderm. And here you can see mesodermal cells migrating from the posterior towards the anterior along what's called the mesodermal wings. So we looked at calcium dynamics of the gastrula. And what we see is that with the onset of gastrulation, calcium oscillations get restricted to this mesodermal population that's been the newly formed mesodermal populations. So the VE and the epiblast and other cell types more or less go silent, whereas the mesodermal cells start showing these oscillations initially closer to the primitive streak, and as they migrate along the mesodermal wings laterally across the embryo. And if we image the same embryo at taking volumes every 10 seconds for several hours, we see around at 3 hours, once these cells have reached the anterior, the oscillations are more enriched in the anterior region. So this shows that the calcium oscillations are not present in all mesodermal cells, but there is a subset of cells, and these cells end up in the anterior of the embryo. And we also quantified this, and we found that as the cells migrate towards the anterior, their frequencies become more regular. Although they're still not in phase with each other, they have more regular frequencies. And one thing we know is that amongst these cells are the progenitors of the heart. So this takes us to the second part of the talk, is to trying to see how coordinated behavior mediated through calcium is active within the developing heart. So far we've been looking at the embryo from the side, where we see the mesodermal cells migrating from the posterior to the anterior, where they meet at the midline to form a structure known as the cardiac crescent, which is the first cardiac structure, recognizable cardiac structure to form in the embryo, but which is visible from the frontal view. And this is a view that we'll be looking at from now on. So I won't go through details of heart development morphogenesis, but I want you to appreciate that the cardiac crescent undergoes a lot of morphogenetic change across a very short window of developmental time. So in the lab, we have developed a way of staging the embryos, with stage 1 defining when we see first visible contractions within the cardiac crescent, and it goes through stage 2 and 3, and just over 12 to 18 hours gives rise to the linear heart tube which is a basic pump, which is the enclosed structure. So I'll be referring to these stages as I talk through the maturing cardiac crescent. So what do we see? So what we see is that as cells of the mesoderm march in to populate the cardiac field, they continue to have these oscillations. And this was in fact described by Richard Tizer and Antonio Miranda when they were in the lab, and they termed it spontaneous asynchronous calcium oscillation, so SACOs for short. So what you see is that these SACOs are present when the cardiac crescent first forms, but just in the matter of few hours, cells learn to coordinate amongst themselves in order to produce this calcium phase wave, which traverses across the entire cardiac field from one side to the other, leading to the contraction of the entire tissue, and if you like, the first heartbeat. So here's another example of the same embryo actually in culture, initially showing SACOs, and now 3 hours later, you see a nice calcium phase wave traversing across the cardiac crescent. So we have still a lot of unanswered questions related to this. For example, how do SACOs become the wave, or is there a conserved direction of the initial wave? And also questions about how is this wave generated and maintained? Because I need to remind that this is at a stage when the heart is the only organ present in a sense. There is no cardiac pacemaker or conductance system, and there's no innervation. So well before there is any differentiation of these structures, you're still getting coordinated activity across the entire cardiac field. So I'll be focusing on these last two questions for the rest of the talk, because the first one is a bit more challenging, and then we're still trying to figure out how this happens and doing a lot of modeling and collaborations with mathematicians and physicists to understand how this could be achieved. Okay, so when another master's student who joined the lab developed a method for determining the direction of the calcium wave in an unbiased manner by quantifying the traces from several movies of calcium waves throughout cardiac crescent development. So just to summarize the data, what we see is that initially at the early stages, we see what's called a asymmetric wave, this wave that goes from one side to the other. And in majority of the cases, these are going from the left to the right. But soon as the cardiac crescent matures, you see symmetric waves on both sides, i.e. Waves that emerge at the lateral aspects of the heart and travel towards the medial. So just to visualize this in the time series, at stage one, what we are seeing are these waves going from one side of the heart to the other, which is what we call an asymmetric wave. This could be perhaps because one side just happens to mature faster than the other, but soon the other side catches up and you see these two independent waves going from the lateral to the medial. Initially, they're out of phase with each other, but as the cardiac crescent matures, they become in phase with each other to form this uniform lateral to medial wave. And with further embryonic development, as the embryo undergoes folding and the formation of the lateral linear heart tube, what you see is its lateral medial wave gets converted into a caudal to rostral wave. So now we have a time series of how this wave evolves through development with the morphogenesis of the heart. But this still leaves us several questions about how the wave is generated and maintained. So be it a asymmetric or a symmetric wave, there is some means by which the wave is, the cells are coordinating and coupling with each other. So in nature, there are two ways in which you can get such coordination. So one system is, the example I'm showing here on the left, is one that's dependent upon what are called local oscillators. So here you see a group of bees that are sending up to a warning signal where you have a wave propagating through the whole swarm. So in this case, there's no individual bee or group of bees that are essential for the wave to exist. So in other words, if you remove the bee or group of bees, the wave's not going to collapse as long as a critical number of bees exist. But in contrast, you have excitatory systems here, which I show through slow time-lapse imaging of lightning strikes, where you can imagine a lightning strike which has an origin, and then the wave travels through space. And there is an excitatory trigger from which needs to be connected to the space in order for it to get excited in one sense. So if you imagine if you take a large pair of scissors and cut the space, there won't be a wave propagating beyond that point because it's separated from the trigger. So this is to distinguish between those two systems. In the case of the cardiac crescent, that's exactly what we did. We took the smallest possible scissors available and then just made a cut in the middle of the cardiac crescent. And what we saw was that if you cut at the midline, the independent waves can exist in the left and the right. However, if we did a trisection of the cardiac crescent, whereby you separated the medial segment from the lateral aspects of the cardiac crescent, the calcium wave in the medial segment completely disappeared. And here are just traces showing that how the medial segment just flatlines after it's separated from the lateral aspects. So this showed that even at these early stages, when there are no cardiac pacemakers or a conductive system or innervation, the wave is still dependent upon excitatory cell populations. And these populations reside within the lateral aspects of the cardiac crescent. So the next step was to try and locate where this trigger population was exactly, so that we could do a molecular characterization of this population and also do functional studies to see if it can act as the pacemaker at these early stages. So what we did was we designed an experiment in which we take an embryo and we characterize the calcium oscillation, so the direction of the calcium wave in the embryo, and then did multiplexed HCR to look at gene expression, focusing on genes that encode proteins that are essential for cardiac pacemaker function or electrical activity of the heart much later on in the mature heart, to see if they're differentially expressed and how it relates to the direction of the excitatory calcium waves. So what we saw in the linear heart tube, for example, was that when the waves are going from the from the caudal to the rostral, a lot of these genes were enriched in the caudal regions of the linear heart tube. But if you looked at embryos that were at earlier stages, when the waves were either going left to right or lateral to medial, what we see is that there is no left-right difference, but the genes that encode channels such as the HCN4, which is the cardiac pacemaker channel, is enriched in the lateral aspects compared to the medial. But we need to still further pinpoint where the excitatory population is so that we can identify unique genes that might be present there, which is essential for defining this population. So what I'm going to present over the next few minutes is just ongoing work in our attempt to try and localize where this population might be so that we can try and do, for example, transcriptomics to try and identify what is unique at the molecular level about this population. So one approach is we can look for regions of differential behavior. For example, here we're looking at the heart at a much later stage. And if we compare, for example, the right ventricle, you see the behavior of calcium is very different. So here you see a wave of calcium that travels from cell to cell that sweeps across the entire field. And along with comes the contraction of the tissue. But in contrast, if we look at the region where you find the sinoatrial node, where the cardiac pacemaker originates, you see that cells initially flicker before it initiates a wave. So there are regions with visible behavioral calcium handling activity that is different based on what function it might be playing. So this is what we're attempting to do in the earlier cardiac crescent stages, to try and locate regions in these lateral aspects of the cardiac crescent where cells might exhibit behavior similar to the sinoatrial node. The other approach is just to chase the wave. It's just to chase the wave to see where it originates from. But this is a very challenging problem because we are trying to image a relatively large structure in 3 dimensions at cellular resolution, at high temporal resolution, while chasing a calcium wave. And there's also the contraction of the cardiac crescent and the distortion of the whole embryo. So it makes it a rather challenging approach to try and exactly see the cell or the cells where the wave originates from. But with lattice light sheet microscopy, again, we are succeeding in kind of trying to address this. For example, with frame rates of about 200 frames per second, we can get image of the wave in 3-dimension at about 3 volumes per second. So at the earlier stages, when the frequency of the wave is a bit slower, this is one approach we are trying to do to try and pinpoint where it originates. So just to summarize the second half of the talk, what I've shown is that there is pattern calcium phase waves, which are established within the heart soon after the cardiac crescent is formed. And these initial calcium phase waves are dependent upon an excitatory mechanism, and the excitatory population lies within the lateral aspects of the cardiac crescent. So what we're trying to do is trying to locate this population so that we can combine, try and see, do lineage studies, for example. Are these the first cells to arrive or the last cells to arrive? Are these the cells that go on to become the sinoatrial node? And what are the molecular characteristics of this cell population? Then also we can do more targeted functional studies, trying to see if, do voltage imaging to see if the excitatory signal originates from the cell population, do ablation experiments to see if it really plays a pacemaker function at these early stages, and so on. So the take-home message is that calcium is important from fertilization onwards, that it plays a lot of important roles in order to initially to drive morphogenetic events, and later on, as the new organs form, to drive the functional maturation of these organs with a special focus on the heart, which is what we study. So this just leaves me to thank members of the lab, of the Srinivas lab, especially Matt, who did a lot of the work that I presented from the pre-implantation embryos, Richard and Antonio, who first identified the sarcos that I described and did the early works on the heart, Konstantinos, who helped with the perigastulation characterization of calcium waves, and Karina, who helped me, a master student who helped me with the later stages, trying to determine the direction of the waves and so on. And also thanks for all the funding that we received from the Wellcome Trust, BHF, and the BPSRC. And also a big thank you for the BSDB for the travel grant, which gave me the opportunity to go and attend the Centenary meeting in Liverpool earlier this year. Thank you.
Thank you very much, Shifaan. That was absolutely beautiful talk. What wonderful movies. So I'm going to start off with a question. I was interested in when you had your cutting experiments, and then you could get these sort of split your oscillator and get these sort of independent oscillation patterns.
Right.
In that instance, you're also causing damage, and injury is also associated with sort of calcium fluxes. So I was wondering, how do you, in your experiment, sort of work out what's injury and what's separation of these two structures?
Yeah, that's a very good question, and that's always a big worry when you're trying to kind of manipulate the embryo mechanically in this sense. So we thought about doing laser cuts as well, but in the end, we realized that actually could cause more damage by just the heat dispersal around the region. So, but just by going mechanically and cutting, we can cause less damage, but you do see calcium accumulation near the cut edge. But what we're looking at is the calcium wave, and it's just numbers as well in the end. So if you see something reproducibly, like doing a cut experiment, and you still see the medial section always kind of going flatlining, then it is a connection with the other sections that is responsible for the oscillations or the wave to exist. So yes, we do always keep it in mind, and sometimes it's not easy because we're just going with our hands to do this cut on a structure that's only 200 microns. And sometimes if you're not happy with the cut, if you think we could damage the embryo too much, we wouldn't include that, because in some cases, if you damage the embryo, the wave completely disappears as well. So when we can do a cut, which we're happy with, which is a clean cut, where the medial segment has been isolated from the lateral, that's what we go and quantify.
Perfect. And we've got a couple of questions from the floor. So question is, in the different tissues you've observed, do you ever find cells that never exhibit calcium activity? If so, are there any correlations with the identity of those cells?
Yeah, that's a very good question. And they are, yes, the answer is yes. Like if you, not all cells of the embryo, even if you go to earlier stages, like just the peri-implantation embryo exhibit these oscillations. So if you, let's say, look at the visceral endoderm, you'll see certain cells that go on and off repeatedly and others which don't undergo oscillations at all. And at these early stages, the frequencies are very variable as well. There are fast oscillations, slow oscillations, and and then it's only with the maturation of the mesoderm you get frequencies to become more regular. So even as I mentioned, even in the mesoderm, it's not all mesodermal cells that undergo oscillations, and even in the cardiac crescent, when we see sarcos, not every single cardiomyocyte is oscillating. But it's hard to see why these cells are different in any sense. So we haven't really looked into that. So when we looked at, let's say, one tissue, we don't know what makes certain cells more prone to oscillations than the other cells. But that's an interesting question that we should pursue, possibly in the early embryo, because there are fewer cells to look at. And
yeah, absolutely, less complex.
Yeah.
So another question, do you predict that the triggers that activate calcium activity are the same, or are there context-specific triggers that differentiate between implantation calcium signaling versus gastrulation, for example?
Yes, I think they're very different. So even the calcium waves, implantation and gastrulation. Okay, so in early development, like from implantation up to gastrulation, we think the mechanism of these underlying oscillations, are the same because we can stop them by targeting the calcium reuptake channels. Whereas in the heart, when sarcos happen, we know that is not the mechanism because it is more dependent on calcium-sodium exchanges and things. So we need a different set of inhibitors to stop the sarcos and the calcium waves. So from implantation to gastrulation, we think the mechanism is the same, but once the mesoderm matures and forms the cardiac crescent and the waves emerge, the mechanism is entirely different.
And I'm going to fit in one last quick question, because you mentioned about ablation. So I was wondering, in that case, what are your plans for the ablation? How many cells do you think are in this little sort of region that's going to be the controller?
We don't know, but just based on the size of the cardiac crescent, I would imagine, I don't know, tens rather than hundreds of cells.
Yeah, so it's relatively localized. Yeah,
that's what we think, because I've done cuts where I can go closer and closer to the lateral, and then to see if we can still sustain some kind of wave. So just based on that, I think it's going to be a small population. But then with ablation, when the cardiac crescent, rest of it is intact, we also have the possibility at the early stages, is there a second population that can mature and take up the function? So these are interesting questions. So is it already very defined, or is there enough plasticity that if they are lost, that something else? Because later on, once you have a cardiac pacemaker in the heart, any damage to the pacemaker has very detrimental effects. But early on, how much plasticity do you have? Can you ablate cells? Maybe the wave will collapse, but then the cells will mature enough to kind of restart the wave. So these are very interesting functional studies that we are hoping to do